In 2008, a Russian archaeologist excavating a cave in the Altai Mountains of Siberia found a small piece of bone, not a skull, not a jaw, a finger bone, roughly the size of a child's fingertip, so fragmentary and so unremarkable that it was catalogued, labeled, and set aside while the team continued working. The cave itself was well known to the scientific community. Denisova Cave had been occupied by humans across multiple periods of prehistory, and the team was not expecting any particular surprise from this particular fragment. 2 years later, when the bone was sent to Svante Pabo's laboratory at the Max Planck Institute in Leipzig for ancient DNA analysis, the result was one of the most extraordinary moments in the history of science. The mitochondrial DNA from that finger bone did not match any known human population. It did not match modern humans. It did not match Neanderthals, whose genome Pabo's team had been working to sequence simultaneously. It was something else. Something that had never been seen before. A population of archaic humans that had left no recognizable fossil record, that had been entirely unknown to science until that moment, identified entirely from a piece of bone smaller than a fingernail. They called them Denisovans, after the cave where the fragment was found. And in the years since that first discovery, what the genome of the Denisovans has revealed has changed not just our understanding of human prehistory in Asia, but our understanding of what it means to be human in the fullest biological sense. The Denisovans present a problem that no other archaic human population has presented quite so starkly. We have their genome before we have their bones. In the normal course of paleoanthropology, a new species or population is identified from fossil remains. The anatomy is described, and then, if conditions permit, DNA is extracted and analyzed. The anatomy tells you what the creature looked like. The DNA tells you how it was related to other populations. With the Denisovans, this sequence was reversed. The DNA came first. The anatomy is still almost entirely unknown. What we have of the Denisovan fossil record is extraordinarily sparse. The original finger bone, a tooth, also from Denisova Cave, a fragment of a skull from the same site, and then, in 2019, a jawbone found by a monk on the Tibetan plateau, recognized as potentially significant, and eventually confirmed through ancient protein analysis as Denisovan, the first Denisovan fossil found outside Siberia. This jaw, known as the Shahe mandible, dated to at least 160,000 years ago and was recovered from an altitude of over 3,000 meters, suggesting that the Denisovans had adapted to high-altitude environments long before any modern human population reached the Tibetan plateau. 4 bones, or more precisely, bone fragments totaling a few inches. That is the entire physical fossil record of a population that, based on the genomic evidence, once ranged across an enormous territory from Siberia to Southeast Asia and may have persisted for hundreds of thousands of years. The story that the genome tells about the Denisovans is several orders of magnitude richer than what the bones alone could have provided. The first and most fundamental finding from the Denisovan genome is that they were a sister group to the Neanderthals. The two populations shared a common ancestor, an archaic human population that diverged from the lineage leading to modern humans roughly 500,000 years ago. The Neanderthals and Denisovans then diverged from each other, with Neanderthals occupying a western range across Europe and Western Asia, while Denisovans occupied an eastern range across Asia. This much was established by the initial 2010 analysis of the finger bone, but the subsequent and even more remarkable finding was what the Denisovan genome looked like when compared not to Neanderthal genomes, but to the genomes of modern humans from different parts of the world. Because when that comparison was made, the Denisovans did not disappear from the human story. They appeared in it, in the genomes of living people, in a pattern that was simultaneously expected and shocking. People of European ancestry carry between 1 and 4% Neanderthal DNA. This finding had already upended the clean replacement model of human evolution. But when researchers looked at the Denisovan contribution to modern human genomes, the signal was different in ways that told a completely different story about the geography of ancient human contact. Modern populations from mainland East Asia, from China and Japan and Korea, carry very little Denisovan ancestry. This was puzzling, given that the Denisovans were an Asian population and might have been expected to have the most contact with the ancestors of modern East Asians. But the puzzlement resolved when researchers looked at populations further east and further south. Aboriginal Australians carry approximately 4 to 6% Denisovan ancestry. Papuans and other Melanesian populations carry similar proportions. Filipino Negrito populations carry even higher proportions, with some studies suggesting up to 8% Denisovan ancestry in certain groups. The pattern of Denisovan ancestry in modern humans does not point toward the Altai Mountains where the fossil was found. It points toward Southeast Asia and the Pacific. The Denisovans who left the largest genetic mark on living human populations were not necessarily the Altai Denisovans. They were a population, or populations, further south and east, who encountered the ancestors of the people who would eventually populate Australia and the Pacific. This finding has profound implications for our understanding of how the Pacific world was populated. The ancestors of Aboriginal Australians and Melanesians were among the earliest modern human migrants out of Africa to reach their current homelands. The timing of their dispersal, somewhere between 50,000 and 65,000 years ago, predates the migrations that produced most East Asian populations. And on their journey from Africa through South Asia and into Southeast Asia and beyond, they encountered Denisovans. Not just in the Altai, but somewhere along the southern route, in the tropical forests of what is now Southeast Asia, or in the island chains that were then connected by lower sea levels into a continuous landmass that geologists call Sundaland. The DNA evidence suggests that these encounters produced offspring that contributed substantially to the founding populations of Australia, New Guinea, and the Pacific Islands. Those contributions are still detectable today. 4 to 8% of the genome of every Aboriginal Australian and every Papuan carrying the molecular signature of encounters with a people who have otherwise left no name, no written record, no monument, no mythology that has survived in any recognizable form. There is a further complication to the Denisovan story that the genomics has revealed in the years since the initial discovery, one that suggests the Denisovans themselves were not a single uniform population. When the Denisovan genomes available for analysis were compared to the modern human populations that carry the highest Denisovan ancestry, the genetic distances between different Denisovan individuals suggested that the Denisovans were internally diverse, with different regional populations showing distinct genetic profiles. The southern Denisovans who contributed ancestry to Australians and Melanesians appear to have been significantly different from the Altai Denisovans, whose fossil material gave the group its name. This means that when we say the Denisovans, we may be referring to what was, in practice, a diverse set of populations spread across a vast geographic range, connected by their shared separation from the Neanderthal lineage, but not necessarily forming a coherent single group in any meaningful cultural or biological sense. The name we have given them, derived from a single Siberian cave, may be as misleading as it is convenient. The high-altitude adaptation story deserves particular attention because it connects the Denisovans directly to the lives of millions of people living today. The Tibetan people have long been recognized as possessing an extraordinary physiological adaptation to high altitude. At elevations where most humans develop altitude sickness, where blood oxygen levels drop dangerously, and the body responds with a series of compensatory mechanisms that are ultimately damaging, Tibetans live and work and thrive. The adaptation is genetic, centered on a variant of the EPAS1 gene that allows the body to respond to low oxygen in a fundamentally different way. When researchers looked at where this EPAS1 variant came from, they found that it was not a recent mutation that had arisen in the Tibetan population after they settled the plateau. It was a Denisovan sequence, inherited from the Denisovans who had been living on the Tibetan plateau for at least 160,000 years, long before any modern human population reached those altitudes. The Shahe mandible, the Tibetan Denisovan jaw dated to at least 160,000 years ago, represented a population that had already solved the high-altitude problem that modern humans would later need to solve. And when modern humans finally reached the plateau, they solved it by the most efficient mechanism available. They interbred with the people who had already figured it out. 30 million Tibetan people living on the roof of the world owe their physiological capacity to thrive at altitude to a genetic variant they inherited from a people who have been otherwise entirely forgotten. The Denisovans did not survive as a distinct population, but in the bodies of Tibetans, in the oxygen-carrying efficiency that makes high-altitude life possible, They are very much present. A 40,000-year-old gift, still working, still essential, still keeping the people who carry it alive. The Denisovan story is, in its way, the purest expression of what the ancient DNA revolution has made possible. Before 2010, the Denisovans did not exist as a scientific concept. They were entirely unknown. The physical fossil record was too sparse and too unremarkable to reveal them. A finger bone from a cave in Siberia, a tooth, a jaw from Tibet. Not enough to build a picture of a people, not enough to give them a name, not enough to place them in the human family tree. The genome did all of that. From a bone smaller than a child's fingernail, it extracted a complete enough picture of a population to place them phylogenetically, to measure their relatedness to Neanderthals and to modern humans, to trace their genetic contributions across the living world, from the Altai to Australia, and to identify specific functional genes that they bequeathed to the people who carry their ancestry today. This is not the archaeology of the future. This is the archaeology of right now. And what it has revealed about the Denisovans will not be the last revelation. More Denisovan fossils will be found. More Denisovan genomes will be sequenced. More living populations will be studied for Denisovan ancestry signals that have not yet been detected. The picture we have now of a diverse and geographically widespread archaic human population that interbred with the ancestors of modern Australians, Melanesians, and Tibetans, and then disappeared, leaving only a fragment of a finger bone in a Siberian cave and 4 to 8% of the genome of people living on the other side of the world, is a beginning, not an ending. There are people alive today who carry more Denisovan DNA than Neanderthal DNA, who carry in their cells the genetic legacy of a people who were entirely unknown to science until 12 years ago, who owe specific physiological capacities to archaic humans they have never heard of, and whose bones we have not yet found. The Denisovans were never what we thought because we never thought about them at all. They existed outside the story. And then a finger bone ended up in a laboratory in Leipzig, and the story had to expand to include them. It has been expanding ever since. Human history did not begin when we started writing it down. It did not begin when we started making tools or burying our dead. It is older and stranger, and more entangled with other kinds of minds and other kinds of people than any story we have previously been able to tell. The Denisovans are the proof of that. A whole people, recovered from a fingertip, still living in the blood of those who followed them. What we know now is a fraction of what the Denisovan story contains. The caves of Southeast Asia, the islands of Wallacea and Sundaland that were connected at low sea levels, the ancient forests that no longer exist in the same form, all of them may hold evidence of a people we can now identify by their genome, but cannot yet see by their bones. Every new excavation in Asia is potentially an encounter with the Denisovans. Every new genome sequenced from a Pacific or Southeast Asian population potentially carries a signal from people who lived in these landscapes before any modern human arrived. We gave them a name from a Siberian cave. They may have been most fully themselves, 10,000 kilometers to the south, in tropical forests and island chains and highland plateaus that the name Denisova does not remotely suggest. The full geography of their world is still to be discovered. The full measure of their contribution to the living human world is still to be calculated. The story is open, the finger bone opened it, and what poured out has not yet stopped flowing.